Bismillah ar-Rahman ar-Rahim. Welcome back, friends. Welcome to another video lecture by Kami Biology. Today in this video lecture we are going to talk about interferon. So let me write. We already discussed the interferon in the cytokine videos lecture on the computer screen, if you guys remember. But in this video I will explain the interferon on diagrammatical level, that how actually the interferon perform their action. Okay, if you guys remember, we talked that interferon are, again, these are what? These are the low molecular weight cytokines. Low molecular weight cytokines. Okay, which are usually produced from the eukaryotic cells. Okay, now this interferon are, whenever this interferon are released against the viruses, then they can facilitate and stimulate and activate our general immune systems. Okay, now second thing is that this interferon is a specific, it is a type of a specific immunity. Okay, now why it is a type of specific immunity? Because it is only released against the viruses. Okay, and whenever there is a virus infection inside our body, then interferon are released in a large number. That's why we can say that it is a specific. It is not released against the bacteria, against the fungi, against the euglena. It is only released against the viruses. That's why we can say that it is a specific immunity. The second thing is that it is also non-specific immunity. Okay, now it is a part of non-specific immunity, as we talk that it is released against the viruses, but it is released against different viruses. Okay, in non-specific immunity, if you guys remember, we talked that it is that type of immunity which is activated against different kinds of the microbes. Similarly, in this case, the interferon is also non-specific immunity because it is activated against different kinds of the viruses. Okay, moreover, we have almost in the previous video lecture, if you guys remember, we talked the interferon classes. So here I will also explain the interferon classes. So let me write interferon classes. Okay, now the first class we have interferon gamma. Interferon, we have interferon gamma. Interferon gamma. Shortly, now this is the sign of interferon gamma. Okay, this interferon gamma contain 20 different families molecules. They contain 20 different class of molecules, okay, which are usually related to the interferon, and these 20 different families of molecules usually secreted from natural killer cell. They are secreted from T cell. Okay, now these 20 different families of interferons will be secreted from the natural killer cell, T cell, and some of the infected cell. Okay, remember, in this case, the interferons, these interferons will be secreted when the cell is infected. When the leukocyte is infected, then such interferons will be secreted. Then we have another class of interferon, we call that interferon beta and alpha. Now interferon beta and alpha are usually secreted from virus infected fibroblast. Virus infected fibroblast. Okay, now what are the function of each interferon? If we explain the function of each interferon through the diagrammatically, so let's suppose we have a cell in this case. Let's suppose. Now this is a cell which is attacked by the viruses. Let's suppose the virus attack that cell. Okay, this viruses also contain their own genetic material. Now these viruses release their genetic material or inject their genetic material inside this leukocyte. Okay, now once they release their genetic material, then after that, due to this genetic material, transcription and translation process happen to this genetic material, and after some times they will release out different kinds of viruses. So we get different kinds of the viruses. Now this is the nucleus of the cells. Now what actually happen? Once we get different kinds of the viruses, before each virus is mature and release out and disturb this cell which is infected, what actually happen? This nucleus contain different kinds of genes. Each genes are activated. Okay, so we get the genes. Now this genes is actually interferon genes. Interferon genes, and they will synthesize to produce interferon. So after some times we will get interferon as you are watching. Now these interferon are released out from this infected cell, and on the other hand we have another cell. Let me draw. Now this is another cell. So these cells will be protected. Whenever this infected cell, let's suppose if this cell is infected, due to this infected cell, the other cell will be protected. Why? Because they release the interferon. Now on the surface of each cell, they contain the receptors. If you guys remember, we already talked about the viruses life cycle. Okay, we talked different viruses life cycle. If you missed that video, it is present into my channel, that how the viruses are synthesized, how the viruses are attacked, how the viruses are attached on the cell. Okay, now what actually happen? Before this virus attack on that cell, the interferon are released out, and the interferon are attaches to each of the receptors at that side of the receptors. Now when the receptors are filled by the interferon molecules, due to this way the viruses are not attached. Okay, the viruses just come here, and this viruses try his best to attach, but the viruses are failed. Okay, now this is the virus. The viruses try to attach on the receptors, because the viruses will inject their genetic material when they attach to the receptor of the host cell. As they are not attached, or there is interferon due to which the viruses are not attached, in this way they will not secrete their genetic material, and due to this way the viruses will not form their copies, and finally after some times these viruses may be die. Now these genes are get activated when the viruses are completed. These genes before not a complete viruses are formed. What actually happen before that, when the virus attack on the cell and when they inject their genome, let's suppose this is the genome. So what happened after that? After that, this genes of the cells of this host cell will get activated, and when they are get activated, then the genes released antiviral drugs. they release antiviral protein. So after that they synthesize the antiviral proteins. Antiviral proteins. Now once this gene synthesize antiviral proteins, now such antiviral proteins are against to this genomes. Sometime they will block this genome. Let's suppose this is the protein, antiviral proteins attached to this one genome. Due to this attachment, the process of the translation and transcription are halted or stopped. Okay, so the process, or we can say a replication process, are stopped. A replication inhibited. Sometime what actually happen, this antiviral protein will cause the destruction of each. Let's suppose we have another genome of the virus. Now sometime this antiviral drugs will cause the destruction of this genome of the viruses. So the viruses genomes are degraded. Okay. So they will also cause the destruction of virus genome. Destruction of virus genome. So that's in a sense about the interferons. I hope you understand about the interferon and the function of the interferon. But if still you have any kind of questions in your mind, you can write it into a comment. Inshallah, I will give you answer as soon as possible. So like this video, share this video, and subscribe the channel to get more interesting videos lecture regarding to the immunology. Thank you so much for watching.